I'd like to thank everybody for coming today. I'm pleased to introduce our speaker for the Genome Center Data Science and Metabolism Group seminar this month, and West Coast Metabolomics Center seminar. So John is got his bachelor's at Rice University, and then walked across the street to Baylor College of Medicine and joined Dr. Bing Zhang's lab about 2 years ago. And he's been the lead software developer on the Web Gestalt, which they just released the Web Gestalt 2024 version, where they've incorporated metabolomics into this pathway enrichment analysis set. So he's here to tell us a little bit about what they do, how this program is working, how to use it, what it can be used for, and what problems we give him, and how maybe we can open a dialogue and improve some communication between the metabolomics and the pathway enrichment software development set. So with that, John, welcome, and looking forward to your talk.
Well, thank you, Dr. Newman, for inviting me to come speak with you all, and thank you for coming. So my name is John Elizarraras, and I'm a bioinformatics programmer in Dr. Bing Zhang's lab at Baylor College of Medicine. And today I will be speaking about one of our most popular tools called Web Gestalt, and the updates I've been working on to improve the speed, but also with support for metabolomics and initial multi-omic integration. And so commonly in a study, you may find that you have a list of genes that are maybe overexpressed in a tumor sample, or you have a list of proteins that are associated with a phenotype of interest. And so when you have this list of genes, you can do a standard association analysis and identify significant proteins, but that alone is not going to be enough to tell a biological story. So this is where enrichment analysis comes in. It allows you to find a meaning from a list of analytes. So for example, from these list of genes, instead of just providing a list you can talk about how these list of genes are supported by are DNA replication related, play a role in chromosome segregation. And so this allows you to, state sort of the biological processes that these, proteins interact in, but also provide statistical backing for it. And it allows you to sort of narrow down your focus. If you're particularly interested in one of these pathways, then you can show that you have some support from your association analysis. So this enrichment analysis has existed for a long time and allows you to create that sort of biological overview from a list of interesting analytes. And in the past, typically this has been with gene and protein data. And you can identify enriched pathways. Maybe you're interested in or different what cellular components your your list of genes participate in. And we've been growing to expand to different phenotypes, cancer associations, ontologies, and other type of analyte sets. And there's a lot of different methods for enrichment analysis. Overrepresentation analysis is the most popular and the most well established. Gene set enrichment analysis and network topology analysis allows you to expand sort of the basic statistical test that ORA provides. I'll go into more detail about the first two. Network topology analysis is sort of probably the newest, and it allows you to sort of do this enrichment analysis, but in a network setting. And so Web Gestalt is a web-based tool that allows you to perform all those three different enrichment analysis methods. It is well established. It's existed in some form since 2005. And so over the years, we've been adding more features, the different analytical methods I mentioned earlier, more analyte sets, and support for multiple ID types, organisms, and custom databases. And so what this sets us apart from other enrichment tools, such as Enricher, is that we allow you to, the support for multiple ID types is unique to us. We provide, you can give us an ID type in any format you want, and we will just map it and do that work for you. The support for multiple organisms, and we have around 80 different databases you can search against per organism, and if we don't have one that you would like, you can always upload your own, and we'll run the statistical analysis for you, but you still have to provide that database. And then, so what we have been adding recently is support for metabolomics, specifically for pathway enrichment. And additionally, to expand on that, we now have the ability to add, like, you can run the enrichment analysis against multiple lists, and we provide, some statistical methods to integrate those lists to, create a sort of combined picture from either, like, you have 3 different ohms you would like to look at, or maybe you have 3 different treatment arms. We allow you to integrate all those data sets together and create like a grand picture to sort of find maybe some agreement across your different like omic types. And we also require no installation. Obviously, this is a website, but for people who are more like technologically inclined, who would like to automate this process, we have multiple software packages and also public APIs. So if you have a website, you can also redirect to our website and automatically populate all the fields, and then you can allow the user to then specify if they're not interested in pathway enrichment, they can, look at the options that we have. So I just wanted to go over like the different types of analysis and sort of like why they're sort of used. So overrepresentation analysis is the most common. It's very simple, both in how you use it and also the output you get. So here it's as simple as you identify a list of interesting genes, for example, and you compare it against a reference list. And the test is a Fisher-exact test comparing the proportion of genes that are present in, for example, a pathway compared to the reference. And the idea is that you're going to be finding pathways or analyte sets that are overrepresented, and that supports the conclusion that your genes are in a certain pathway. And so the input is just going to be a list of interesting analytes, and then you have to provide a reference list, which is sort of the background that you're comparing against. And this is especially a problem for metabolomics. For RNA and protein data, your coverage is relatively high. You can probably go for just all protein coding genes, and you're good with RNA data. And we provide a lot of default reference lists, but for metabolomics data, we found that identification varies from study to study, and so it's really for overrepresentation analysis specifically. For metabolomics data, we recommend that you provide that reference list by yourself. But for RNA and gene data, we have, we already have predefined reference lists that are good enough to use for that. And also there's a lot of caveats with overrepresentation analysis. It's very sensitive to annotation coverage. You should have a relatively good understanding of the database that you're looking against. Like if you're using wiki pathways, you should understand how that's done and how well annotated that pathway is, because that can have a large effect on your final results. And then finally, as I mentioned previously, that reference list selection is very important, especially for metabolomics, where your identification usually is really low, and you're dealing with like even your reference list is probably like in the 500, like maybe 500 to 1,000 genes versus RNA and protein data, you're in like maybe like 10,000. So the difference in size really is really important for overrepresentation analysis. But the output here is that you're going to get a lot of enriched analyte sets, hopefully, if your results are good, with different scores. So when for overrepresentation analysis in WebGestalt, we provide interactive tables, bar charts, and a lot of other visualizations that let you explore the data deeper. A lot of other tools will just give you this, just a bar graph, and then allow you, and then just list all the analyte sets, but they don't let you explore the specific details. Like, for example, this DNA replication that I mentioned earlier, you can look at the actual intersection and understand what genes are actually participating in that DNA replication. So that's, and then for ontologies, we also provide, a ontological view. And if you click on these links, you can explore deeper into like what that actual like Go term for this example is, and what is the actual explanation of it. And then so gene set enrichment analysis is sort of the second most popular method that we have. And this really tries to combat all the issues that you will see with overrepresentation analysis. Requires a little bit more work and a little bit more insight from the user, which is why it's not used as often. But here, instead of just giving, deciding what analytes are interesting versus what was your background, you're just going to give gene set enrichment analysis everything. So all your identifications, and you also going to be providing a ranking metric, which is going to depend on what type of biological question you're asking. So for us, commonly we run in association with tumor versus normal samples. And so we would have protein, you would have an analyte and then a sort of ranking metric that is its association with this with a tumor. So commonly it's just negative log 10p, sine, log, fold change. There's a lot of different ranking metrics you can choose. You can just do the z-score of its abundance. But here, the idea is that you're going to be sorting a list where the genes at the top of the list are going to be maybe overexpressed in tumor versus underexpressed in tumor. And so the method here will for each pathway, it'll calculate if this gene is in the pathway, add this ranking metric to it. If not, then subtract it. And it allows you to identify enriched analyte sets, but on top of that, it lets you indicate the direction, which is the overrepresentation. You just see it's enriched, but you don't know, maybe it's overexpressed in tumor versus underexpressed, and that is something you won't get with overrepresentation. But with gene set enrichment, you can actually see the direction. So if you see a negative enrichment score, that indicates that the genes that participate in that pathway are located near the bottom of the list. And so depending on that ranking metric, you can then come to a bigger biological conclusion. And so some other things to note for people using gene set enrichment is that for the background for p-value calculation, it does a lot of random permutations. And so that means, depending on your list size, you're going to get different results every time you run it. Especially for metabolomics, again, I'm going to keep harping on how tough metabolomics is. If your list is very small, like your identifications are low, just because of the random chance you're seeing, you're going to get a lot of different results. So that's something to pay attention to. But again, the key point here is that you're going to be able to see pathways that are upregulated versus downregulated. So you can see that this is an example from just a tumor study we see, and you can see a lot of these immune response pathways are downregulated, as you might expect, versus you can, there's not a lot of upregulation, and even, some of the pathways that you see, are not very significant. But you come to the conclusion that you see a lot of immune response, and you can also note that it's downregulated. Which is the key point here. And then there's a lot more robustness with gene set enrichment. You're not running into the issues about coverage as much as you do with overrepresentation. And so this is neat to see just a bar graph, but for people, if you want to tell the story, and you're probably going to be not interested in just showing this bar graph and letting someone else come to having to read all these options and decide what pathway is important. So usually this, the user will identify maybe they're interested in one specific pathway. So we allow you to visualize on the actual pathway map the results. So here is an example from this pathway in Wiki Pathways, and then all the proteins are actually colored by the ranking metric that you see. So you can see that which proteins are downregulated. And so in this pathway, that you can see that all of them are downregulated, and you may see that maybe some proteins are up versus down. So this allows you to visually see the actual response in the pathway. And so WebGestalt supports this coloring in Wiki Pathways and KEGG figures, both for RNA, protein, and also metabolomics. Yeah. So that's has been how the tool has been used. Usually, we've been focused on gene and RNA data and protein data, but we wanted to expand as we see that metabolomics is becoming more involved in sort of the studies that we saw, and also is growing in a lot of different ways. But before we wanted to expand and add more features to WebGestalt, we wanted to improve the performance. So that was actually the first part I worked on. And then some of the other things that we worked on is, again, the addition of metabolomics. And then as we have a lot of databases across that support RNA and protein, we wanted to look at ways that we can combine all these different omic types together and create a sort of a multi-omic view of pathway enrichment. And then to support all that new integration, we also created new visualizations to support users understanding the data that they get out from WebGestalt. And so I would love to talk about this a lot. I'm going to go a little broad here, but one of the things how we address the performance issue is that we created a new Rust core that powers all the packages that we work on. And this was done because the amount of data that we process is increasing every year. And as we know, for multi-omic integration, we're going to be processing at least 3 times the amount of data that we would normally. And so we wanted to make sure that our server can achieve the performance that we would like, because as we have more users, we don't want them to be waiting like 5 minutes. Usually enrichment analysis is something very quick. You just want to get a quick answers. So that's why we worked on the performance. And so this new Rust library is used to power the actual like computation of all the software packages. So the web server itself uses our R package for performing enrichment analysis, but because everything is done almost all in Rust, we can actually use it across different platforms. So we have the R package that has existed for a long time, but now we have a Python package for people who would like to use Python for enrichment analysis. And the library is open source, and so more people can integrate it very easily if they would like to use it in their own tools. But overall, we found that the performance increased by like 90%, which allows us to process 20 times more data than we could in the previous update. So overrepresentation analysis is usually very quick. Even in the old version, it took like milliseconds to run. But gene set enrichment analysis is very computationally expensive, as you have 1,000 different permutations you have to calculate through. So that took almost like 5 minutes to run. And now we get that down to 12 seconds. And this is allows us to run a lot more samples all at the same time. And then network topology analysis is very similar to overrepresentation, but before it does overrepresentation analysis, it does this random walk. And so that process took a long time as well. So that normally would take 3 minutes, and now we get that down to 17 seconds. And so by making Rust do all the hard computation, we allowed it to increase performance. But the benefit is that none of the actual results from the end users change at all. So the users will have won't know that this performance, they'll have no effects on the actual statistical output. So this benefit is just for free essentially. There's no algorithmic change that we have to do for optimization. It's just just porting it over to a different language. And so for people on the server, they get the speed benefit, but also for people who are using our software packages, you will get these same numbers as well. And so that was the first major hurdle that we achieved with the new Web Gestalt version. And it was sort of a cakewalk compared to metabolomics support. So how Web Gestalt typically works, how it has been built in the past, is that we have all these different ID types that we would like to support. And so in for protein data, we support 80 different ID types. And it's really well established. This gene maps to this gene in a different database. And so we have a lot of well-established mapping tables, and allows us to create this sort of standard ID. So we can map all these different ID types to one ID that is then used for the actual enrichment analysis, and it allows us to output the results. And so we really need this standard ID, but as sort of I'm alluding to, metabolomics really struggles with this. Pathway databases like KEGG, Wikipathways, they all use their own unique identifiers. So we would have to map that to some universal ID if we want to support this ID mapping that Web Gestalt has. But even in these pathway databases, they actually provide their own mapping tables. So in Wikipathways, they say this metabolite is equal to this metabolite. But then you look in the different mapping table and they disagree. Or you may have multiple ID types that map to the same metabolite. Or you may have two of the same ID types mapping to 2 different metabolites. And it is a really big struggle for us because in enrichment analysis, we work in absolute terms. It's this gene is in this set or it's not. Or this metabolite is in this set or it's not. So this was a big issue for our tool moving forward. Luckily for us, someone else has already done this work in terms of this metabolite mapping issue. So we've used RampDB, which is a project coming out of the NIH. And the main issue, what their main task here is to just take all these different metabolite ID types, and these are machine readable IDs. These are not just regular names. And so the disagreements between them is very vast and very complicated. So they created an automated pipeline to sort of solve this issue. I would recommend reading the paper if you're very interested in this. A very broad strokes sort of workflow that they look at is if they see that 2 ID types they see that one ID type maps to 2 different metabolites, or there's some like conflicts. They run it through this sort of conflict resolution pipeline where they check the variance in molecular weight, and if they find a large enough difference, they'll manually verify all these ID types, and then determine whether or not they should be merged to a single ID. And so RampDB itself has thousands of standardized IDs, and they support 14 different ID types. The most popular, of course, being PubChem, KEBI, HNDB. There's a lot in KEG. So users can use any ID type that they would like, and then we can then perform this conflict resolution from the ramp by using the RAMP database. And so that allows us to solve this issue that we saw with this sort of conflicting ID types. And then for the metabolites, we have stuck currently with pathway enrichment. So we support KEG, HNDB, Wikipathways, and Reactome. And then so this is something that we would definitely like to improve. Currently, we can tell you enrich pathways, but there's also other interesting aspects of metabolites you may want to be interested in, such as the chemical classes they belong to, and other things. But for metabolomics, this ID mapping issue was a big problem for us, and we'd love to talk about how other people have handled this. But even if we have those mapping tables, when we were looking for examples to sort of test how our tool would run, metabolomic data just comes in a lot of different forms. Sometimes you have no machine readable IDs, right? It's just a list of metabolites with just their names, and those naming conventions they may have IUPAC names, they may have a 3-letter abbreviation, they may have a full expanded name, and for us we can't really use that because there's those names. How do we know what you mean? That's a lot of intervention has to be done by the user at that point. And also we find that sometimes metabolite data, they just give you IDs without any annotation about what source it comes from. So this is an example from a real study I found, publication, where they just gave you metabolites with numeric IDs. And then I looked, there's no annotation. I couldn't tell, is this PubChem? Because PubChem is numerical. KEBI is also very similar. And so that's another problem. We can't really solve this naming convention, as that is very specific. But for us, how we try to sort of alleviate this issue is that whenever we output any metabolite names in the tables that you see in your results, we standardize as much as we could to the RefMet names, because in RAMP they do provide metabolite names that we can provide, but again they also have this issue that the namings are all very different. So as much as we can in the output from Gestalt, we try to make it so you can reference some source for understanding where that metabolite name came from. And then, so for user convenience and also sort of solve this unlabeled issue, in WebGestalt, you just give us the list of IDs. You don't need to tell us what ID type it is. We'll just detect it for you. And so for metabolic data, we only support 14, but again, for there's sometimes you have numerical data, and it's very hard for users who are not very well informed how to perform analysis or what ID type it is. So we'll tell you what that ID type is. And also for gene and protein, this came in handy because there's 80 ID types to select from, which is really difficult. So also for people who don't understand their own gene data or what ID type, maybe they got data from someone else, they don't know what ID type it is, they can just give it to us and we'll tell you exactly what ID type it is. So we have this metabolite data. We have support for protein, RNA, and then the question becomes, you can perform individual analysis and you can get results. You can find, in RNA data we see these pathways are enriched, and protein, these different pathways are enriched. But we would like to sort of create a more holistic picture and how we analyze multiple omic types together. And so then you can also, for example, if you have, you're running a study with multiple treatment arms, you may be interested in seeing what pathways are commonly observed in all of these 3 different treatment arms. So it doesn't have to be a mixture of omic types. You do perform a meta-analysis using the same omic type. But the process for integration is the same for both of these methods. And so how WebGestalt handles this integration is that we perform the enrichment analysis for each or each list separately. So it's going to be the exact same method as if you ran that list individually. And so to create that integration, how we did that is we calculated meta p-value, which is where we take the p-values for each of the pathways for each individual ohm. And there's a lot of different methods to do that. We support two. Most common is Stouffer, where you basically sum up the z-values. You convert the p-values to z-values, and you can add them up. And you can get like sort of this meta p-value that represents sort of how the agreement of statistical significance across these three different lists. And so on the right is an example of the output from Gestalt. So here is just the output from all three different lists. But for people, you may also be interested, you can also click on these tabs and view the individual results from RNA, protein, and metabolite data to observe the differences. Because commonly, RNA and protein may have disagreements in what pathways they think are enriched, and the metabolites are their own world sometimes. So that's something also you might want to be interested in. And so this enrichment analysis is sort of like this meta p-value is sort of how it has been historically done. And since Web Gestalt itself we try to make it very easy to use and understand. This is sort of where we ended with multi-omic integration, but it still allows you to sort of observe interesting trends. And so to sort of give an application of, and to sort of show how this works, this is an example that you can actually use. If you go to webgestalt.org, we have examples, and this is one of the data sets you can just load if you're interested in learning how to use the tool. So this is just a study that was collected RNA, protein, and metabolite data from a lot of from HeLa cells. And for us, the nice thing was that their RNA, protein, and metabolite data was really good. So that's why we used it. But the findings that we would like to see here are mitochondrial-related pathways. And so the idea here is to sort of see, can we use metabolomics by itself to sort of observe these mitochondrial pathways? In here, they treated these cells with FCCP, which is a drug that disrupts the mitochondrial membrane potential. So the idea is that you're probably going to be disrupting any mitochondrial-related pathway. And so how typically we would run overrepresentation analysis for this example as we calculate a p-value for the difference in abundance between the two different conditions between control and then the treated samples. And then I took all the metabolites where the FDR is less than 0.01. So these are where significantly different metabolites. So that's our interesting list. And so then and then all the metabolites, including these interesting metabolites, are sort of the background list in the overrepresentation analysis. And so that's sort of typical workflow for overrepresentation analysis and how you'd use our tool. And so if you run that, if we run it with wiki pathways, you can see that you actually do get three statistically significant pathways. Amino acid, you do get some mitochondria-related pathways. But something to note here, especially with metabolomics, is that you're going to get a lot of pathways that are very general. This is mainly due to annotation. Wiki pathways and KEGG, their metabolomic support is very low in comparison to protein and gene and RNA. In total, there's about like 2,000 annotated metabolites that we support versus RNA and protein, you're like 10,000. So the difference is very large. So that's something to consider when you're doing an analysis. But you can still see that you can still get statistically significant pathways. And if you actually look at the amino acid pathway, again, it's very large. But you can see that these dark blue boxes here are the metabolites that were highly abundant. Or had a very significant difference between the two different control groups. So again, you can visualize where in the pathway you see it, and you can draw some new conclusions. And it can draw your attention to certain regions where you do have a lot of one for metabolomics data, you have a lot of these metabolites are highly abundant enough that you can identify them, but also that their abundance is significantly different between the two different groups. And so. It's just sort of a reminder. In the middle here, we see the TCA cycle, so that's also very important, and will come up for our multi-omic integration. So for us, we saw that this TCA cycle, there's a lot of metabolites here that are highly different. So can we use the protein? Does the protein and RNA data support this TCA cycle? And also, what other conclusions can we get by integrating the protein and RNA data from this study? And so, but before we get into that, if we go back to this, the results that we get, again, there's only 3 enriched pathways. When you actually look at the protein data that we did when we wanted this integration, you can see that the difference is very stark. You get a lot more enrichment, and also the pathways you get are more specific, also very related to the mitochondrial. So this sort of indicates, that metabolomics on its own by enrichment may not give you the exact conclusions that you need or would like. And additionally, if you look at KEGG, you get nothing, which is great. And this is mainly caused by how KEGG annotates it. As you saw previously, we had amino acid metabolism. A lot of these are just amino acid metabolism, but they're just broken into specific amino acids. And so again, as you remember from earlier in the slide, like how the coverage of your pathway the pathway database you're looking at is going to change your overrepresentation analysis results. So that is something else to consider with metabolomics. And if we look at metabolomics in the reference set that you choose, also has a large difference. So if we look at that KEGG result that I did previously, if you use all the annotated metabolites that you can see in KEGG, which is how RNA and protein data is done. Usually you just use everything that's identified in the pathway database. You're going to get a lot of enriched pathways, right? So you're getting 42 pathways that are enriched significantly. Versus if you narrow it down to, only looking at the experimental, identified metabolites, you're going to reduce the amount of pathways that are enriched, but these typically will be more like real pathways. And then when you look at GSCA, which I would say is the gold standard in terms of metabolomics data, you can see that you also get a reduction when you consider GSCA. And so for metabolomics specifically, we really encourage you to only include what you actually identify. We actually have a warning when you run metabolomics. And this sort of a warning label for metabolomics and overrepresentation analysis is very finicky. There's a lot, but I would say a large part of that is not driven by the statistical method. It's just based off annotations in pathway databases. And so as time goes on, and I think as people, work on, metabolomics in annotation, this problem will be solved in the future. But in its early stages, this is something that we came across that sort of differs from protein and RNA data. And so we see that metabolites, you get 2 enriched pathways. Some of them are, maybe they're somewhat interesting, but we also see that we have RNA and protein data. So how can we integrate all of that together and create, a better understanding? And so for the workflow here is very similar. We're going to be doing GSCA to sort of solve that issue that we saw previously. So for each omic type, we're going to calculate a p-value for a difference in abundance, the same as we do for representation. But instead of creating a threshold of what we consider interesting, we're just going to include everything. And then the rate metric I used is this negative log 10p sign fold change. But again, there's different rate metrics you can use, but this is the most common, and it allows us to sort of separate our list into proteins that are highly expressed in the treated cells versus control. And so the idea is then, can we use the multi-omic integration with GSCA to identify pathways that are affected by the treatment with this drug that disrupts the mitochondrial membrane potential. So when you run it, you actually get a lot of enrichment. This is the combined view of using all the data together. You get a lot of pathways. And so that's sort of the benefit. Is you actually get a lot more statistically significant results. And a lot of these are, you see the oxidative phosphorylation, mitochondrial complexes, and then you also can see some of the pathways that are being supported by metabolomics that would not be significant on its own. So that sort of shows the importance of including all the data in here. If you ran these samples individually, you would not see a lot of these pathways as enriched. And so you can see that that you can find and identify the mitochondrial pathways. So if you're running the study before coming to any conclusions, it would lead you to the direction of exploring, maybe I need to understand the mitochondrial, like what pathways they participate in. And so that's a really, where I would say enrichment analysis shines is if you're not sure what direction to look towards, you can quickly find pathways, or maybe you're interested in biological processes that these enriched genes or these abundant genes participate in, and that can lead you to further exploration. And so again, if we look at the visualization of multi-omic pathways, this is sort of a new visualization that we created. Instead of having one color, you can see in each of these genes, we actually have the left. It's split in half, where the left side is the RNA data, and on the right is the protein data. And you can see in this pathway, which is the Krebs cycle, which as you saw in the previous with overrepresentation analysis with metabolomics, you can see that all these pathways are actually associated with the control, which would make sense if you believe that that this pathway is disrupted by the fact that it was treated with a drug that just completely disrupts the mitochondria. And something interesting that you would not see if you ran these results initially is that you actually see disagreement between these different between RNA and protein data, which is interesting because you would assume that there's may be agreement, or you may see that there's some agreement with RNA and protein, but then you see some certain genes you have disagreement between whether or not it's associated with the control or with the treated samples. And so this is again, we support this multicoloring in KEGG and wiki pathways, and we support as many lists as you want, and you can see the difference in the associations for each of these different lists, and it allows you to quickly visualize the different how the different omic types sort of agree or disagree with each other. And so a question that we came towards is that we can do this enrichment analysis. We can find these pathways, and then we can say, we can explore the Krebs cycle. That's very important. But it's sort of the next steps that are less clear. We see certain genes that there's agreement, some where protein RNA disagree, and then you have where maybe the protein's higher, and then you can have the opposite effect where the protein's lower. And so some of the further things that would be a potential further direction is, can we actually find meaning in this discordance between omic types? There's a lot of information that because in enrichment analysis, is it in the pathway or is it not? And that's really where it ends. It allows you to explore, identify pathways that you would be interested in exploring, but it's not going to create the final conclusion of why this is important. But it can identify maybe directions to look forward to. And additionally, for pathway enrichment specifically, there's a lot more work you can do. Right, reactions have directions. You have substrate, the enzyme, and the product. In pathway enrichment, again, it's just, is it in there or is it not? And it'd be interesting to see if you see the substrate going up, the protein going up. You should expect maybe there's a relationship to the product in that relationship, and so that's not going to be that's not going to be captured in enrichment analysis, and so there's a lot more work to be done. We think again, WebGestalt in itself is supposed to be like a quick exploration tool, identify pathways that you may find interesting, but this is, I think, a further direction for people who are interested in this multi-omic integration. Again, you can find enriched pathways, and maybe it can inspire you to, look further into these disagreements or the reactions. And so, metabolomics analysis is, I would say, the largest, most difficult part of this of the new update. Again, as I mentioned earlier, I think for people for enrichment analysis people, the standardization of metabolite IDs and names is a big struggle. There's a lot of uncertainty, as I think a lot of people here know. For people who would like, if you're publishing data sets, for one request I would have is at least have one machine readable ID for each metabolite, even if they're mixed. If you can't find that PubChem has its metabolite, at least give us an HMDB ID, because if you publish it, we're lacking. We're not going to be public. We may not have access to all the information that we need to resolve what metabolite you're actually talking about. And something else that we would like to work on is that the naming scheme it'd be nice for us to just import names and do that and tell you what ID type it is. But the naming scheme is very complex. So I would say for just saving your metabolites in their names is not enough for other people to be able to use the data in an efficient way if you're looking for enrichment analysis or integrating it with different omic types. And so that's sort of a request I have for as someone who's working on this tool. But something else that I think will definitely be done in the future is that there needs to be more complete pathway databases. And Wiki Pathways, which is I would say the best annotated for metabolomics, there's 484 Wiki Pathways that have at least one metabolite in it versus 845 that have genes. And so when you're doing integration, the amount you can do is limited by how many pathways that actually support metabolites. And so I think over time, this will improve. But if you have pathways that you're interested in, Wiki Pathways allows you to just submit them, and so we can submit them, and it will be reviewed and added to WebGestalt and all these other different enrichment tools. But the annotation for metabolites is definitely lacking. Again, there's like 2,000 annotated metabolites that participate in pathways across all the different pathway databases that we could find, versus genes and proteins, you're looking in tens of thousands. And one of the additional problems is that in the metabolite data sets that I've come across, like after WebGestalt, there's a lot of unknown metabolites. And there's a lot of information that I think that could be gained from this. So for enrichment analysis, again, we're absolutes, either in the pathway or it's not. We don't do any similarity comparison for in gene and protein data. So for us, we sort of we kept that formula for metabolites, but there's a lot of unknowns. And so all that information is going to be, in its current form in WebGestalt, is currently lost. And so there's a lot of information that we think we can gain from that. But that is a question that for us, we're not metabolomics field, and I think there's a lot of work that can be done to address this. So I'm very interested in other people's ideas about how do we deal with these unknowns and not just disregard them. For us, we've seen that even 75% of metabolites we have no ID for, but we still have associations. We still have their measurements in all these different samples, and we can see that these unknowns are actually very important for tumor versus normal comparisons. But we just have no way to sort of understand what they are in the first place, which would be great because we can't just put in a study, unknown 004 is very important in breast cancer. It'd be great, but that it's not very useful. So that's something that is identification, but also even if we couldn't, directly use the metabolite name, we can still maybe identify, this metabolite is probably related to this pathway or is part of this, chemical class, and then we can go on from there. So I think there's a lot of work that we can do with these unknown metabolites. And I would say for people, for the current field of enrichment, I think this is where a lot of work could be done from people that we have not talked to yet. I think Enricher is a really popular one. They don't even touch metabolites at all. I think we're probably one of the first to do it. So from our perspective, I think we could learn a lot from you guys about how you guys handle your data, how well your IDs are. Do you guys want support for multiple ID types? Like for us, it's usually all gene symbol or all KEGG IDs, but sometimes you may have metabolites that don't have any PubChem annotation, but some others do, so you have a mixture of IDs. So these are a lot of features that we would like to collaborate on. And I think generally, the biggest area for improvement is sort of this unknown metabolites and sort of how do we expand on that. And so for us, how we've currently have planned for WebGestalt in the future is that we want to add more organisms for metabolites. Currently, we only support humans, mainly because that's all RAMP has provided. So either if we want to add more organisms, we're going to have to create our own metabolite mapping system or copy what RAMP has done and replicate that for different organisms. And additionally, all the visualizations that we have on WebGestalt currently is built mainly for gene and protein data, where it's but for metabolites, you may have different needs. So that's something else that we were looking at, is improving the visualizations that we have. And then something that's unique for metabolites. Currently in WebGestalt, for how it has been done, proteins, people don't have disagreements. They will trust however you map your IDs, versus for metabolites, that's going to be a different issue. Especially since RAMP in itself is very sort of complex about how it handles these merging of IDs. So it's, how we better communicate that to users is something that we're also going to be improving. So for example, you may input a list of 500 metabolites, but then WebGestalt will tell you, actually, we found that these are really there's only 386 unique metabolites in this list. So that's not, it'll tell you how many metabolites we actually resolve these to, but I think we could better communicate like how that process was handled, allowing for mixed ID types, etc. And there's a lot of different ways we can handle ID mapping, which I've learned in my past day that I've been here. The problems seem even greater than I realized. Sort of shocking coming from, it's a culture shock, I guess, coming from the proteomics world. Yeah. And then, as I mentioned previously, the multi-omic integration that we have currently is, it's what has been done, it's well established and has been published, and a lot of people have done this in studies to do this meta-analysis, calculate the meta-Ps, and, find this agreement. But I think, the current way it works is that we're most of the time it's going to be searching for agreement, where you see, all three of these omic types all agree that this pathway is important, but maybe it would be interesting to look at where they disagree, or again, capturing the more complex relationships in the pathways, finding why, the relationship between metabolites and proteins versus, the disagreement between RNA and protein. And finally, one of the last things that we would like to do is, if we create this, improvement in multi-omic integration, the better visualization of these combined results. Because currently we can show the visualization for, pathways. It's very easy to visualize that, but there's a lot of other analyte sets that we support in WebGestalt, such as Go terms, phenotype associations, and also like single cell, like what type of cells commonly express, these type of genes. So there's a lot more work that we can be doing in that area as well. And so I would encourage you guys to try us out if you are interested in WebGestalt. You can go to webgestalt.org. For people who are interested in, doing this in a more automatic fashion, there's a link to see all the different packages. So we currently have R and Python. And we really encourage people to, if you are missing a functional database that you would like to add, maybe like there's chemical classes that you would like to be added, like please talk to us. We would like to add as much as we can. We also, if you have your own like database that you would like to add to WebGestalt, we have a specific category for community contributed databases. But so please let us know what we could add. Metabolomics itself is a very new field for us. We sort of just touch the surface of using the same type of techniques that we see in gene and protein data and applying that to metabolomics. But again, there's still a lot more work that we can be doing in that area. And again, for people who are interested in using the tool, there's a lot of examples you can look at for both the single list and also the multi-list. So hopefully it's very easy to use. Please check us out if you're interested. And if you have any questions, you can always, if you go to the user forum, I'm going to be the person responding to you. So any questions you have, please let me know. I would like to thank everyone for coming, and specifically the people in my lab, all the people in my lab back at Baylor College of Medicine. There's a lot of people who worked on this tool. It's been existing since 2005. Yuxing is another bioinformatics programmer in the lab who did a lot of the grunt work in terms of the previous versions. And also our collaborators in Gladstone, Alex Pico and Christina Hanspers, who are leading the Wiki Pathways effort and have sort of helped us with the visualizations of the pathways that you saw in this presentation. Thank you for coming. That's all I have. If you guys have any questions or any conversations you'd like to have, please let me know.
Thank you. Very interesting talk. Enlightening. So one of the problems of metabolites is that you work to try to map them to biochemical pathways, which is one way you can categorize metabolites, but likely that's not the most important thing. much more important is what metabolites do. So the functional set enrichment, being like inflammatory, anti-inflammatory, a drug, a food compound, like functional. So there's a database called ChemPhont, the Chemical Functional Ontology. You might want to look at it.
Yeah.
So they have a very large classification system that is much more function-oriented than biochemical pathways, which are ignoring transporters by and large. Because if you find, say, malic acid, you don't know if it's a mitochondria. It maps to your TCA cycle, but it actually might be the real dominant thing is in the cytosol, right? Which has a very different function. Same for citrate and whatnot. So I really would encourage you to kind of think about that more functional-oriented view that you also do for gene set enrichments, like cell cycle replication, whatnot, rather than a biochemical linear thinking. Second, one problem in metabolomics is we don't have a reference set, as you said. And so your solution is to say people should provide a reference set. The reason why you say that is because you use Fisher exact test.
Yep.
Don't use Fisher exact test. So you want to look at apply the Kolmorova-Smirnov test, which is independent from the size of your set. We published that 8 years ago or so. So that we showed it that you cannot use Fisher exact test in metabolomics.
Yeah.
That's why. So these are just two comments. Oh, the third question now. Now comes the question. What about lipids? I mean, many of us just love lipids. And many of us, especially in Davis, think lipids are like the center of the universe. Can you tell us about lipids?
I will say that in terms of like imitation, again, because we're sort of branching out from what we have currently in WebGestalt. We haven't branched into lipids. Again, this is something that we would like to, of course, expand into. In terms of us, I think we started with what's familiar and what we could prove is what we know. So if you could, we would like to explore lipids, but in terms of what type of sets would be interesting is something that we personally have not much experience in. So if you could, point out what type of you give a list of lipids, what outputs would you like? What annotations would you like to see? That's the question. Yeah.
We can work on that.
Yeah. Do you have? Is there
Well,
yeah.
if we would know, we would have made it.
Yeah,
right?
yeah. That's the issue. Is we have to have
But we get
annotations
all the time when we present statistical results to people in funky terms, in chemical class enrichment. That's what we usually do.
Yeah.
And we say the lysophosphorylethanolamines are really upregulated, and then they say, and what? And I'm like, well, that's very significant. And they say, and? It's like, well, I need a WebGestalt for that to give us more function of this, right?
Yeah.
And we don't have that tool, so
That's good. Yeah, and I'll look at the two resources you mentioned earlier.
Any other questions out there? If not, I've got a question or two. Yeah.
I have two questions.
Yeah.
My first question is the user forum link your preferred method of communication if we have issues or feature requests?
Yes. Yeah. So it's that's the preferred form. Yeah.
Okay, cool. And my second question, I was wondering about data privacy for people who work in private sector and have intellectual property concerns.
Yep.
When data is uploaded to the web server, is it retained or does it disappear after a certain period of time?
It definitely disappears after a certain period of time. We run out of storage frequently, so we delete it. But if you are worried about uploading to somewhere else, if you use software packages, all your data won't be. It just downloads it from the server, so you don't have to worry about it being sent somewhere else. If you would rather avoid using web server at all, you can use the software. WebGestalt R would do the exact same thing that the web server does. It just requires a little bit more coding. Yeah.
Okay, thank you.
All right, so I had a couple little questions. I didn't notice PathBank. Do you have PathBank in your database searches? which is what HMDBs seem to map to.
We have the small molecule pathway database, but PathBank, I don't think.
Yeah, I think that's it.
Yeah, that's the same thing. Yeah.
Yeah.
I think they use a lot of different names. Even the
Right.
metabolite database names are
Yeah.
different names.
So
Yeah.
one thing at least in HMDB, there's the cascading ontology.
Mhm.
So I'm wondering whether or not you can search back like higher up in the tree.
Yeah, that's actually something that we definitely can do. Gene ontology is an example of an ontological system that's really popular for protein and gene enrichment, and we have a lot of ways to visualize the ontology, but also we have different ways of resolving redundant sets and find hierarchical structures and determining where in the ontology there's a greater source of
Okay.
enrichment. Yeah.
So then, second question was, metabolites participate in multiple pathways.
Yeah.
So how do you deal with that problem?
I think my answer would be that we don't.
Okay.
Maybe in terms of what type, depends on your biological question, because the idea for us, the enrichment analysis is just to point out these pathways are significant, or they you have a lot of all of the pathways, a lot of the metabolites you gave us are present in these pathways, and so that's sort of a next step of now you see these two pathways are both enriched, and you see that there's a lot, maybe there's some shared metabolites, but that's would be sort of a next step would be one, enrichment analysis would be identifying that pathway for you, and then you can then ask those questions of which ones are shared. But it would be interesting to highlight maybe the shared, how the different pathways intersect with each other.
That's kind of what I was thinking as far as visualization and importance, because that could point towards systems biology, functional change like Oliver's looking at.
Yep. That's a good idea.
Next question I had was, where did it go? Oh, so something you said made me start thinking, and something I've seen in certain data sets. Do you think you could apply some kind of network analysis to essentially do a bottleneck analysis in this? So you could see, essentially highlight points in metabolism where you might have a blockade, where you're getting a buildup and a deficit that cascades then through metabolism? Yeah, I'll let you try and answer that vague question.
No, that is sort of touching on that idea of terms of future directions of capturing that relationship between reactions and also for GSA we will have that information of what's up and down. So that is definitely possible. That would be, we do have network-based analysis methods, but that you're also going to consider the actual if it's up or down. That's something that we could adapt with existing methods too, and there's definitely a lot of.
I might have a data set you could play with,
Yeah.
actually. So we should talk again
Yeah.
on the way to the airport.
Yeah.
And then I was wondering whether you could essentially, from the unknown question, would there be a simple way to essentially piggyback an analysis that would allow unknown associations with known pathways somehow? I mean, in a way, it's just cluster analysis, but to be able to say these unknowns found within this data set or across these multiple studies always seem to change when this metabolite subset changes. So do you think that this approach could be co-opted for some analysis of that sort?
Yeah, definitely. Yeah, I definitely think you could. You would have to make some slight modifications on how they handle, because for enrichment analysis, how it's currently based is each entity is separate, but you could treat a cluster as a single entity and then resolve where that entity, if you see that a lot of the proteins or a lot of the metabolites in that cluster participate in a pathway, you can sort of assign that cluster to that pathway and then do the same type of enrichment analysis that you see here.
Right. Okay. Thanks.
Unless somebody else.
I don't see a hand. Oh, wait, I have a hand.
How does it identify the pathways, for the application? What's the confidence level that this is the right pathway?
Yeah.
That you want to observe?
Let me go back and so we can show the plot. Hopefully there's a smaller one. But I'll go to for overrepresentation analysis. It's a Fisher exact test, which is just, it gives you the p-value. Like how overrepresentation analysis is usually plotted is not actually by the, unfortunately I can't zoom in, but so here it's actually by the enrichment ratio, which is just basically like this is 1.6 times more like enriched. But there is FDR control that you can see, like these dark blue plots here are going to be like FDR less than 0.05. And then for GSCA, there's also the p-value in FDR. So that's how it typically is done. GSCA, like instead of doing a Fisher exact test, it does the same type of, it calculates an enrichment score using like a running sum method, which is a little outside the scope, I think, of this presentation, but essentially it does that exact same thing for 1,000 random permutations, and then it uses that to calculate the p-value based off like how many of those permutations had an even higher enrichment score than your real set. And that also creates a problem of it's random, which means that you can get different results every single time. So you can be like, oh, I found that this pathway is really enriched, and then you show someone else, and you run it again, and it's gone. Which
It's not like 100%. Like it's like, is it 100%, or can you 100% like say like this is the right path?
You could say what you would say is that maybe for this example, you could say like in this protein example, the oxidative phosphorylation is significantly enriched. In fact, it's like it's like 0.00 like e to the -16 enriched, which is really high. But you can also, like, if you click on this like bar and it's on the website, you could actually see what proteins actually participate in it. If you're 100% sure that these proteins participate in it, and you're like almost 100%, you're like 99.999% sure that this is enriched more than what you would expect from like random. Yeah. Does that answer your question?
Yeah. So I really love open access software, open code software, open everything, public, academia, love it. But there's also the corporate side. So there's something called Ingenuity Pathway Analysis, IPA. How do you compare, how do you rate that competition? What does IPA do better than you guys, or where you think you are better than IPA?
I personally haven't heard of IPA. Do you know, like?
Oh. Ingenuity Pathway.
I can't afford it, so I just know that
it's very expensive. Well, first of all,
maybe that's why I haven't heard. I don't think
it's corporate. It's very expensive.
Yeah.
And like people in corporate always use it. Right?
Yeah.
And then people in academia, not so much. Because we afford. But you know, they of course have 25 years of work on it, right?
Yeah.
So, well, okay then. Yeah.
Yeah, but I would say, I have never seen that used. I would say GSCA is like what's most commonly used, but like in terms of comparison, that's sort of just, maybe it's just which one gives you results you like more. But that's not a great, there's no quantitative metric to do that, yeah.
Okay.
Sorry.
What's the current support for PTM analysis, and like can that also be integrated into the multi-omics?
Oh, that's a good question. Yeah, so we do support PTMs. Like you provide us like the sequence and the location of the site, but we only have like two or three databases we support. That is actually something that our lab is also very interested in. But in terms of like in WebGestalt currently, there's no like integration. What you could possibly do, what we have done in the past, is just find the protein that the site is located on, and then put it into WebGestalt as like a protein, and then you could do it that way. But in terms of like, there's a lot more work that we're working on to actually how to use the site information to, drive something. But for now, the easiest way would be remove the site information, resolve it up to the protein, and then give it for enrichment analysis if you want to do a multi-omic comparison.
And for the non-proteomics people, that PTM is post-translational modifications. Yeah. Question?
Yeah.
Thank you for your talk. So I have a question. So in metabolomics, we can annotate a lot of like compounds from environment, for example, explosive compounds. And those compounds are not in like a conventional database, like pathway database. So what is the best strategy to use those compounds anyway in the pathway analysis? It's kind of similar to, I'm not sure if in genomics or proteomics you can see a certain sequence that haven't been curated to a database, and how to use those sequences.
That is a good question. In terms of enrichment analysis, again, like if it's not seen in the pathway, you'll just discard it. I think that like using like outside information is something that I would say like you would have to require like more like a different type of algorithm or like extra steps to apply to this. But if you like the easiest way, if you actually want to use enrichment analysis, the way you have to do it is you have to like create an analyte set of saying these analytes are annotated to this function. So that would be the way you have to use if you want to use WebGestalt or any enrichment analysis tool. You have to have an annotation and all the lists of analytes that participate in it. But in terms of exploring these external ones, I think that would be outside the scope of like enrichment analysis. It's enrichment analysis like is the main tool is like creating the biological context, allowing people to explore like a quick meaning from a list of analytes. And with statistical backing, but that is an interesting point and something I think would be interesting to explore.
Thank you.
Okay, well, I think that's enough in the hot seat. Thank you, John, and thank you everybody for coming.